Welcome to Nikolai's Genetics Lessons, and today's lecture is going to be very interesting. Today we are going to talk about RNA folding. Here on this picture you see two-dimensional representation of RNA folding, but it may take much more complex forms when we are talking about three-dimensional form of the RNA. And also, as you see, this is ribosome, which consists of two subunits, large ribosomal subunit and small ribosomal subunit, which consist of proteins and also RNA, so-called non-coding RNA. But which types of RNA we have? It can be messenger RNA. Its function to provide genetic code for protein sequence building. So here you see messenger RNA here in the middle between two subunits. Also it can be tRNA, you see it also here, which brings amino acid to growing polypeptide chain. It can be ribosomal RNA, which we find in the ribosome in both subunits. For example, in humans, the 40S small ribosomal subunit contains about 33 proteins and 18S rRNA. It is about 1,900 nucleotides. And the large or 60S ribosomal subunit contains about 49 proteins and 3 ribosomal RNAs. 5S about 120 nucleotides, 5.8S about 160 nucleotides, and 28S, the length is 4,700 nucleotides. So in large subunit we can find 3 RNA chains and about 49 different proteins which aggregate in single large subunit. And here you see the structure of single rRNA. So as you see, it is very complex, and you probably have an impression that only super powerful computers can predict how it's going to fold. But actually on the next slide I will going to show you that even you can predict the structure of the RNA using simple methods. Just wait a couple more minutes. And another type of RNA is snRNA, or small nuclear RNA, and other catalytic RNA. They are used by organisms. We can find them in the nucleus, as you see. They just help to process messenger RNA, pre-mRNA, in order for it to be processed to mature RNA, which afterwards would be used to provide a code sequence for tRNA to bring certain amino acids, which corresponds with the sequence of the messenger RNA. Now let's go over simple two-dimensional structure of the RNA, so you would know all the related terminology. So what we call hairpin loop, it is a structure shown here. Internal loop is going to be structure here. Next, bulge loop. This is structure here. I do not spend too much time to explain because I think it is self-explanatory. Next, multibranched loop. It is shown here on this graph because this loop has multiple branches. So 3 branches or more. Next is stem. So there are multiple of them here. So for example, here, what we call stem, here, and here, and here. And the large structure, pseudoknot, is a structure which you see here. And here is another hairpin loop, which I missed. So now I hope you are familiar with terminology, and we can move to the next slide. But before, take a look at this picture. This is 16S ribosomal subunit in the prokaryotes. And as you see, how complex a structure of the ribosomal RNA can be. And still, we can predict its structure. And on the next slide, I'm going to explain how to do it. Very simple method. Maybe you won't be able to do something this complex, but something this complex you would be able easily predict. I hope you recognize this structure. This is tRNA. It has 5' end, 3' end. It is made of single RNA which folds on itself. And this is amino acid attachment site. And here is a simplified two-dimensional model. This is relatively simple model, not so many nucleotides here, so we can easily predict it using, long story short, using dot plot method. If you earlier watched my also other videos about dot plot method for DNA, where we plot one DNA strand on one side, another on the other side, and we plot dots where we have the same sequence in the same place on both strands, excluding noise. Here it's a little bit different. Take a look. Let's say we already know the structure, and let's say, of course, we don't know if we do the first time research. So let's say this is 5' end and this is 3' end, and our sequence starts with UGC. So now let's take a closer look at what we have here. And here's our sequence, UGC. And what the sequence here we have, it is the same sequence. So from top to the bottom, the same sequence as here from left to right. And whenever we have not the same letter, we put black dot. But when we have a pair, we know that adenine base pairs with uracil and cytosine base pairs with guanine. Hence, we put a black dot here. So that means that here our single, not two strands of the RNA, but single strand of RNA is going to be self-complementary. So would adhere to itself. So let me bring the picture back to the scale. Now, how we build this square. So here is going to be beginning, and here is the end of the sequence. So 5' end here and 3' end here. Then another strand, and we are going to have 5' end here and 3' end here. Again, we build another sequence here, which is going to be 5' end to 3' end. I don't have much space left, but here's going to be another sequence with 5' end to 3' end. In double-stranded DNA, we know that adenine base pairs with thymine and guanine base pairs with cytosine. But because we have RNA here, not DNA, instead of this thymine, we are going to have uracil. So adenine base pairs with uracil and guanine with cytosine. And instead of double-stranded DNA, we have here single-stranded RNA, which is going to fold on itself. Any RNA has beginning and has the end. Every run of dots here represent a stem. So for example, here is going to be one stem, 1, 2, 3, 4, 5, and it corresponds with this stem here. Another one is going to be, let's count, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. And this stretch corresponds with this stem here. Another stem is going to correspond with this fragment. And the last one corresponds with this small stem here. And by the way, what this line means, this basically means that we have uracil here and here, cytosine here and here, guanine here and here. So it's just a line which shows where all the nucleotides are the same. And what this picture above this line means is just a reflection of what we have below this line. So we are actually not interested in this part of this picture, only in this part, or we only can work with the top part. It doesn't matter, but it tells us how many stems we have here, their length, and their sequence. And it is now very easy for us to predict two-dimensional structure of the RNA. Hopefully now you have much better understanding how dot plot method works in order to help us to predict RNA three-dimensional and two-dimensional structure. And this is all for today. Subscribe and see you in the next video. Goodbye.